Hello, in this chapter, we're going to be looking at development and heredity. I'm going to divide this into at least two parts. The first part will look at human development from fertilization to birth. The first thing that we're going to talk about is fertilization. Fertilization is defined as the fusion of the sperm and oocytes, haploid nuclei, to form a diploid cell called the zygote. So the sperm and the oocyte, remember, each of them has half the number of chromosomes that would normally exist in the rest of the cells in your body. So you have 46 chromosomes in each cell in your body, but the sperm and the egg each have 23 chromosomes. Whenever their nuclei fuse, it creates a single cell, as you can see here, with 46 chromosomes because they combine their 23 chromosomes together to make 46. Here you can actually see some sperm trying to penetrate the barrier surrounding an egg or secondary oocyte. Now we're going to talk about a couple of things related to fertilization, such as where does it happen? When is it most likely to occur? So in other words, when is a female most likely to get pregnant during her menstrual cycle? And also we'll talk about what happens when the sperm actually meets the egg. So what are the actual steps necessary for fertilization to happen? So where exactly does fertilization occur? Fertilization occurs in the distal end of the uterine tube. So just again to review the anatomy of the female reproductive system, this is the vagina down here, or the vaginal canal. This is the cervix, which is the opening to the uterus. Here's the uterus, and here is the uterine tube, and here is the ovaries. The oocyte is ovulated from the ovaries, and then it must enter the uterine tube. However, it takes the oocyte 72 hours to make its way down the entire uterine tube to the uterus. However, it must be fertilized within 24 hours after being ovulated or else it dies. And this is why fertilization must happen in the uterine tube, because after 24 hours, it's only made it probably about this far. And so the sperm must travel all the way up through the uterus and down the uterine tube in order to fertilize the egg. Next, we're going to answer the question, when is fertilization most likely to occur during a female's reproductive cycle? But before we can answer that question, you have to understand a little bit about the behavior of sperm once it enters the female reproductive system. So during ejaculation, there's about 200 to 300 million sperm that enter the vagina. However, only hundreds make it to the uterine tube where the oocyte is waiting. A lot of the other sperm may get lost. They might go down the wrong uterine tube. Some of them might never ever make it that far, and some of them might actually drain out through the vagina. A really important characteristic of sperm are that sperm are unable to penetrate the egg immediately after ejaculation. In other words, they require some sort of priming, and we call this capacitation. Capacitation is defined as a chemical process that occurs when the sperm mixes with chemicals from the uterus, and it actually makes the sperm capable of penetrating the egg. And so capacitation pretty much does two different things to the sperm. One, it increases the sperm's motility so that it can swim faster. Second, it thins the membrane at the sperm head here. So it's thinning this membrane so it's easier for the sperm to release their acrosomal enzymes. Now remember, the acrosomal enzymes are digestive enzymes that are present in this vesicular sac located in the head of the sperm. And this little vesicle is called the acrosome. It contains digestive enzymes that are released whenever the sperm contacts the egg in order to help penetrate the barriers surrounding the egg. This purple structure here is simply just the nucleus. And then we have a mid-piece here filled with mitochondria to provide ATP or energy so the sperm can stay motile for a long time. And then we also have a tail here to allow for motility. So now let's answer the question, when is fertilization most likely to occur? Let's compile the information that we need in order to calculate the fertilization window. First, you have to remember that sperm can survive 3 to 5 days in the uterus. Second, it's important to remember that the egg is only viable up to 24 hours after ovulation. And third, After ejaculation, capacitation must take place in order for the sperm to be capable of penetrating the egg, and this usually takes about 10 hours to complete after ejaculation. So with all that information and knowing that ovulation tends to happen in the middle of the reproductive cycle, so usually the reproductive cycle is 28 days, the monthly cycle for a female, and around day 14 is usually when ovulation happens. So where would be the best window for fertilization to occur based on this information? Well, if you have intercourse 3 to 5 days before ovulating and within 14 hours after ovulating, that is the best window for fertilization. Now, you can't really go more than 14 hours after ovulation because it doesn't give the sperm enough time to undergo capacitation before the egg dies. This also means that intercourse more than a day after ovulation is very unlikely to end in fertilization. Now, if you're trying to avoid getting pregnant, you're probably going to want to widen this window a little bit because it is possible for some people's sperm to actually survive longer than 5 days within the uterus. And in addition, not everybody's timing of ovulation is the same, and the timing of capacitation might vary from individual to individual. But this also just demonstrates how sometimes when people are trying to get pregnant, especially when they're using an ovulation kit or something like that, if they're waiting too long after they ovulate, then there's almost no chance to get pregnant. You have a higher chance of getting pregnant if you actually can predict what day you're going to ovulate and then you have intercourse a couple days before that because the sperm will stick around for a little bit. This also means that if you think about it, so let's say 5 days before ovulation happens at day 4. So 5 days before that would be like day 9 of your menstrual cycle. Most females will have their period for like maybe 3 to 7 days. So let's just say you're somebody that has a longer period. It lasts like 5 to 7 days or something. That means that if you have sex like 3 or 4 days after your period ends, you could still get pregnant because the sperm could be around whenever you end up ovulating. So what actually happens when the sperm meets the egg? Once the egg is reached, the sperm must make it through 3 barriers that are surrounding the egg. The outermost barrier is called the corona radiata, and it's shown here in this image. It's a layer of cells, actually called granulosa cells, surrounding the oocyte, and these are derived from the follicular cells. So remember, the oocytes were developing in a sphere of cells called follicles within the ovaries. So some of those follicle cells actually stayed attached to the oocyte after ovulation. In some animals, we know the corona radiata actually secretes chemicals to attract sperm. However, this hasn't quite been demonstrated in humans yet. One way that I actually remember that the corona radiata is the outermost barrier around the oocyte is that the term corona is used to describe something that surrounds an object, like the corona around the sun during a total solar eclipse. I actually had the opportunity to see and experience a total solar eclipse when I was living in Missouri. And if you would like to see a video of that, I'll put a link to us watching the total solar eclipse below. It was one of the most amazing experiences I've ever had in my life. The second layer that the sperm have to get through is called the zona pellucida. This is a gel-like barrier beneath the corona radiata. And you can see that right here. And then, of course, it has to make it through the cell membrane. So that's the innermost barrier right here. So just a little bit of a review of sperm morphology. Again, one of the most important structures in the sperm to allow it to penetrate the egg is called the acrosome, which is this cap-like vesicle located in the tip of the head of the sperm. And it contains a whole bunch of lysosomal enzymes, so digestive enzymes that help to break down the barriers around the egg. Whenever a sperm releases these digestive enzymes onto the surface of the egg, we call that an acrosomal reaction. And it will actually allow them to penetrate the corona radiata as well as the zona pellucida. Now let's look at the 4 steps necessary for fertilization to occur. These steps follow a very logical sequence. The first step is known as corona radiata penetration, and this is when the sperm squeezes through the cell layers of the corona radiata. So here we can see the sperm trying to penetrate the corona radiata. Inside here we have the secondary oocyte. It's arrested in meiosis II, so it hasn't quite finished cell division yet. Here you can actually see one of the polar bodies that has been created. So remember, polar bodies are cells that are created during whenever the eggs are forming, and these polar bodies are pretty much just created to remove genetic information so we can create a haploid cell. The polar bodies will eventually die. So after the sperm penetrate the corona radiata, next we have zona pellucida penetration. So remember, the zona pellucida is this gel-like barrier around the egg. And so when the sperm contact the zona pellucida, it will activate the acrosomal reaction where they'll release their digestive enzymes. After the sperm have made it through the zona pellucida, then we'll have one sperm that will fuse with the membrane of the oocyte. So we have sperm and oocyte membrane fusion. So here's the cell membrane of the sperm, and it's fusing with the cell membrane of the egg. Once one sperm fuses with the cell membrane, all others are blocked from entry. So there are chemical changes that happen in the egg that pretty much create this permanent barrier that doesn't allow any other sperm to come through. Once the sperm and oocyte's membranes fuse, next the sperm and oocyte's nuclei will fuse. So an important point here is that only the sperm nucleus enters the oocyte, as you can see here. The rest of the sperm, including its mitochondria, are destroyed. So it's only their genetic information that's being contributed to the egg. And here you can actually see the nucleus of the sperm and the egg fusing together to combine their genetic information. Out here you can see two polar bodies, which are just going to degenerate and die. So one common misconception that I hear is that it only takes one sperm to fertilize an egg, and this isn't necessarily true. If you think about it, hundreds of sperm are actually needed to release digestive enzymes and break down the corona radiata and zona pellucida. So the first sperm that actually reaches the oocyte is almost never the one that fertilizes it. So instead, all these sperm are fighting to get through and are helping to degrade the barriers around the egg, and then one of them will be the lucky one to actually penetrate the whole way through. One clinical application associated with fertilization is in vitro fertilization, or IVF. IVF is a process of fertilization where an egg is combined with a sperm outside of the body, and then the embryo is implanted into the uterus. How this procedure is done is the female is usually given some sort of medication, usually containing hormones. It stimulates the ovaries to produce multiple mature oocytes. And then they will actually remove some of the eggs from the ovaries. Then they'll mix those eggs with sperm outside in some sort of test tube. And then once the embryo has started to divide and it reached about the 8-cell stage, they'll surgically implant one or two of these into the inner lining of the uterus and hope that one of them takes and starts to develop. The rest of this lecture is going to be looking at prenatal development, or in other words, the developmental stages that occur before birth. Embryology is actually the study of prenatal development. Another term that you should know is the term conceptus, and this is just a general term used for both the embryo and the fetus. So it's pretty much a term used to describe everything after the zygote, or in other words, everything after fertilization has occurred. I will use the word conceptus a lot as I describe the different developmental stages. One thing I would like to point out is that we've come a very far way in understanding prenatal development. It was only a little over 300 years ago that it was thought that there was a tiny human living inside the sperm that grew into a larger form. So people used to think that really the man was the only one contributing anything to the new human, and the female was just a vesicle to carry that new human. We have come a long way since then, but there's still so much to learn, especially about how gene regulation causes cells to become specialized during development. It's just crazy to think about all of us starting life as a single cell called a zygote, and from that single cell, as it divides, some of those cells will become specialized to become heart cells. Some of them will become specialized to become brain cells. Some of them will become part of the skeleton, part of the muscles. So what changes are happening in those cells to allow them or to cause them to take these different routes? And so these are combinations of chemical communication within the cell as well as changes in gene expression, because every cell is genetically identical to each other. It's just certain genes are turned on or off. So if we can understand that, then we can gain a much better understanding of how cells become specialized. So now let's look at the stages of prenatal development. Now we can define these stages clinically or biologically, and you want to be able to define them both clinically as well as biologically, and also know what characteristics define each stage of prenatal development and understand the timing of these different stages. Let's start off by talking about the clinical stages of prenatal development. Clinically, prenatal development can be divided into 3 trimesters. The first trimester begins on the first day of the last period, and it continues to the end of week 12. This is when the conceptus is most vulnerable, with more than 50% of them dying during the first trimester. This occurs because the conceptus is very vulnerable to malnutrition during this time, drugs, as well as medications, but also if there's any type of genetic abnormality within the conceptus, it's usually during this time that will be detected, and oh, I'm sorry, I got distracted. Hey, come here. Come here. I'll introduce you to somebody that wants to say hi. Come here, Phoenix. Hey. This is our new little baby, Phoenix. He's only 7 weeks old. Say hi to everybody. Now he's demanding attention. Oh yeah, I love you. Love you. All right, where were we? Another reason that the conceptus is very vulnerable during this first trimester is this is usually where genetic abnormalities are detected. So if there's a genetic abnormality that would prevent the fetus from surviving, this is usually when a miscarriage will occur. During the second trimester, this lasts from the beginning of week 13 to the end of week 24. At week 24, that's the earliest that an infant has a chance to survive outside of the womb. However, it would require significant amounts of medical care in order for that infant to survive. And even with significant amounts of medical care, usually only about 20 to 45% of infants will survive at that stage. The third trimester begins at week 25 and it ends at week 40 or whenever birth occurs. A baby's due date is actually calculated as 40 weeks after the woman's first day of her last period. So if you want to calculate your due date when you become pregnant, you have to think about when was the first day of your last period and then go out 40 weeks from then. Now, this 40-week mark is really just a suggestion because the baby could actually come anywhere between 37 weeks and 42 weeks, and that's still considered normal and full term. So there is a pretty wide window there of when birth could actually happen. The third trimester is pretty much just dominated by growth and maturation of the internal organs to a point where the infant could survive outside of the mother's body. So now let's look at the biological stages of prenatal development, which are summarized in this diagram here. One thing I want to point out is that the clinical stages began with the first day of the female's last period, whereas the biological stages begin with fertilization. So about 2 weeks after the clinical stages would begin. The pre-embryonic stage is the first stage, and it begins with fertilization and continues to about day 16. During the pre-embryonic period, it's dominated by mitotic divisions or cell divisions, which we call cleavage, as well as implantation. So the pre-embryonic period ends after implantation has occurred into the uterine lining. The embryonic stage begins at day 16 and it continues until about week 8. And during this period of time, the conceptus is called an embryo. And the embryonic stage begins when the 3 primary germ layers form, and that is through a process called gastrulation, which we'll talk about later. Another thing that characterizes the embryonic stage is that all organs develop into their basic form during this stage. However, one thing I need to point out is that these organs are still mostly non-functional, so the embryo would not be able to survive outside of the mother's body at this point. The fetal period begins at week 9 and continues until week 38 or until birth. And this is whenever the conceptus is referred to as a fetus. This is dominated by growth as well as maturation of the tissues and organs to a point where the fetus could actually survive outside of the mother. So now we're going to look at each of the biological stages in more detail to talk about what is actually happening during each one of these stages. Let's begin with the pre-embryonic period, which again is like the first 2 weeks of development after fertilization. So it goes up to about day 16 or so. The pre-embryonic stage begins with the zygote. And after we have the zygote undergoes cleavage as it moves down the uterine tube. Now, cleavage is just a series of mitotic divisions, so a series of cell divisions where we're just pretty much cloning the cells. The cells are increasing in number, but not necessarily in size. So you have this one cluster and it just keeps splitting into multiple smaller cells. So cleavage starts out with a 2-cell stage because the zygote splits into 2. And then it becomes a 4-cell stage, and then 8-cell stage, 16, and so forth. Each one of the new cells that are made are called blastomeres. And these are really important because these are stem cells, meaning that each one of these cells could become any type of cell at this point. One other thing I want to point out is that the zona pellucida is still present. That gel-like barrier is still present around this developing pre-embryo. After the cleavage stage, we have what's called the morula, and this is a solid cluster of cells, and it usually begins with 16 cells and can grow up to about 100 cells. This cluster of cells is still free-floating in the uterus and has not implanted yet, and so it's still making its way down the uterine tube. After the morula, we have what is called the blastocyst, and the blastocyst is actually a fluid-filled ball of cells, so you can see that there's all this fluid inside of there. At the end of the first week after fertilization, the blastocyst begins to implant within the endometrium or the inner lining of the uterus. And this process actually takes about a week for the blastocyst to become fully implanted within the uterine lining. So let's look a little bit more at the characteristics of a blastocyst here. So the early blastocyst has the zona pellucida around it. However, that starts to degenerate. And the late blastocyst no longer has the zona pellucida around it, that gel-like barrier. The blastocyst itself consists of a blastocele, which is this fluid-filled cavity in here, as well as this outer layer of cells called the trophoblast. The trophoblast, shown in orange in this diagram, is extremely important because, for one, it secretes HCG. If you remember, HCG stands for human chorionic gonadotrophin, and it is a hormone that helps to sustain pregnancy. It keeps the corpus luteum around so that it can continue to produce progesterone. So this HCG is essential for sustaining early pregnancy. Second, the trophoblast becomes a fetal part of the placenta called the chorion. And we'll talk a little bit more about the structure of the placenta later. But the placenta has a fetal part and a maternal part. And the fetal part is derived from these trophoblast cells. The only structure left within the blastocyst that we did not talk about is this little cluster inside of here. This is called the embryoblast. And this inner cell mass actually becomes the embryo. So the embryoblast cells are considered pluripotent stem cells because each one of these cells has this potential to differentiate into any cell type in the human body at this point. It's only pluripotent for a few days though, because eventually it's going to undergo some types of specialization where some of these cells will start differentiating and taking different paths. This diagram highlights the different parts of a human blastocyst 5 days after fertilization. So here you can see the zona pellucida, trophoblast, blastocele, embryoblast, and then all of them highlighted. Again, let's go through the pre-embryonic stages of development. After the zygote, we have the 2-cell stage, 4-cell stage, and 8-cell stage. So this is all occurring in the uterine tube as this cluster of cells is moving towards the uterus. Once it's reached the uterus, but still free floating, it's called the morula, and that's just a cluster of cells, and it can have anywhere from 16 to about 100 cells or so. This morula, this solid cluster of cells, will eventually turn into what's called the blastocyst, where it has a fluid-filled cavity called the blastocele, surrounded by cells called the trophoblast, which will make HCG, as well as become the fetal part of the placenta. And then this inner cell mass in here is also called the embryoblast and will create the embryo itself. I will put a link to this video below. It actually shows the cells dividing and going through these major stages shown here in this image. One clinical application associated with the pre-embryonic stage are these embryonic stem cells. So remember the blastomeres that are formed through cleavage, as well as these embryoblast cells, are considered stem cells. And so they can actually create all the different cells in the human body. This is really important because research on stem cells help us understand how cells differentiate, or in other words, become specialized to form all the different cells in your body. In addition, stem cells can be used to repair damaged tissues and treat diseases. If you would like to learn a little bit more about how embryonic stem cells can be used in medicine, this is a very short TED-Ed video that discusses the use of embryonic stem cells, and I'll put a link to this video below. Another clinical application associated with the early stages of development is the production of twins. There's actually two main methods for producing twins. The first one is called dizygotic twins or fraternal twins. Now, this occurs whenever 2 eggs are ovulated simultaneously from the ovaries. So normally only 1 egg is ovulated from the ovaries at a time, but there are cases where multiple eggs may become ovulated and actually enter the uterine tube. And in this case, each egg is fertilized by a separate sperm. And because we're starting out with 2 completely different eggs, 2 completely different sperm, these fraternal twins are no more related than regular siblings. Furthermore, each implants separately into the uterine lining, so each will have its own placenta during development. The second method of producing twins is called monozygotic twins or identical twins. This occurs because the early cell cluster, the morula here, or the embryoblast actually splits into 2 separate clusters of cells, and each one develops into its own individual. Because monozygotic twins are derived from a single fertilized egg, from a single sperm and a single egg fusing, they are genetically identical to each other. It's so interesting to think that each one of these cells theoretically could grow into a separate individual at these early stages of development. In the case of monozygotic twins, they may share the same placenta or they may have separate placentas. So here we can see dizygotic twins developing within the uterus, and each one has its separate placenta because they implanted separately into the uterine wall. So here's the placenta is more like a disc-shaped tissue. So here's the placenta for this infant here, and then here's the placenta for the other one. Each one has its own amniotic sac though. Monozygotic twins are shown over here on the right, and you can see that each one has its own amniotic sac, but they're sharing one placenta to nourish both of them. So now understanding how monozygotic and dizygotic twins develop, what do you think might cause conjoined twins to develop? This actually happens because, for example, the embryoblast might start to split into 2 separate clusters, but it doesn't completely divide into 2 separate clusters. Instead, some of the cells stay connected to each other. And so that would lead to conjoined twins without complete separate bodies. So the pre-embryonic stage ends with implantation. So let's talk a little bit about the timing of this event and where exactly it occurs. So let's step back a little bit. So here's the unfertilized oocyte here. It becomes fertilized, remember, in the distal end of the uterine tube. and then it undergoes multiple stages of cleavage. Once it becomes the morula, it will enter the uterus. Then it becomes the blastocyst. Remember that sphere of cells here. And it's actually the blastocyst that's going to implant into the endometrium of the uterus. The blastocyst begins implanting into the endometrium of the uterus about 7 days after conception or after fertilization. So that means it takes about 7 days or so for the conceptus to make its way from here all the way down to the uterus where it's going to start to implant. Then it takes about a week for the blastocyst to completely implant and burrow itself into the uterine lining. One thing I want to point out is that 50 to 75% of blastocysts fail to implant. And this can lead to what is called a chemical pregnancy, or in other words, a positive pregnancy test followed by menstruation and no actual pregnancy. Now, this occurs because the blastocyst does start to implant and it does start secreting HCG from the trophoblast. And remember, HCG is the hormone detected by pregnancy tests. So as this blastocyst starts to implant, it starts to secrete HCG. And if you take a pregnancy test before your missed period, you might actually get a positive pregnancy test. And then a couple of days later, you'll get your period and not actually be pregnant. So that's why it's recommended that you wait until you miss your period before you take a pregnancy test, because there's a good chance that you might just be having a chemical pregnancy. Where exactly does implantation occur? So the blastocyst implants itself into the endometrium, or the inner lining of the uterus. And so here's the endometrium, here's the uterine cavity, and the blastocyst will burrow its way completely into the endometrium, and the embryo will start to develop completely within the lining of the uterus. So where's the most common site of implantation in the uterus? Well, most commonly, the blastocyst will implant in the posterior wall of the uterus. So remember, the uterus usually tilts over the bladder. So this is a cadaver image. This is the bladder here. Here is the uterus. Here is the vaginal canal right here. And so you can see the uterus is tilted over the bladder. So this would be the posterior wall, or the rear wall. And so the placenta will usually grow back on this wall. However, it is not uncommon for the blastocyst to actually implant in the anterior wall. For example, I'm 39 weeks pregnant now, and actually I have an anterior placenta. So that means that the blastocyst implanted in the anterior wall of the uterus. Really, there's not much of a difference if it implants in the posterior wall versus the anterior wall. One of the major differences is that usually if the placenta is growing in the anterior wall, it might be harder to actually detect movements of the infant because you have the placenta kind of blocking the movements. One clinical application associated with implantation is what's known as an ectopic pregnancy, and I mentioned this in a previous lecture video. 1 in 300 pregnancies result in the blastocyst implanting somewhere other than the uterus itself, and this is what is called an ectopic pregnancy. The most common place for an ectopic pregnancy is within the uterine tubes. So the fertilized egg doesn't make it all the way to the uterus and it implants too early. This could happen because of some sort of blockage or scarring in the uterine tubes. In the case of a tubal pregnancy, that would end in a miscarriage, or it would have to end in termination of the pregnancy because it is extremely dangerous for the mother if the uterine tube actually bursts open. Another place where an ectopic pregnancy can occur is within the abdominal cavity, so actually out here. So somehow the blastocyst completely escapes the uterus and it will implant in the abdominal wall or on the surface of the intestines or something like that. Pretty much the blastocyst can start to develop anywhere where there's a lot of blood supply. An abdominal pregnancy is also extremely dangerous. Can lead to hemorrhaging and so forth. There's no way to vaginally deliver a fetus that's developing in the abdominal cavity. So again, this you will usually, if it's detected early enough, will be terminated to protect the mother as well as the fetus. So the normal location of implantation and the development of the placenta would be somewhere on the wall of the uterus, and it would have to be away from where the cervix is, because in order for delivery to happen, the baby needs access to the cervix to exit. There are some cases, though, where the implantation happens too close to the cervix, and this is known as placenta previa. And in this case, this can be very dangerous because it can lead to hemorrhaging or bleeding, and it's very difficult to deliver the fetus in this case because there's no way for the fetus to actually push its way through the cervix because the placenta is in the way. So this baby would probably have to be delivered through a cesarean section. So one last thing I want to mention about the pre-embryonic stage is what's happening to the embryoblast during implantation. So remember, the embryoblast started out as this cluster of cells, but during implantation, it actually forms a 2-layer disc. And it forms 2 layers of cells called the epiblast, which you can see here and here, as well as the hypoblast as the other layer of cells. The epiblast cells are going to be really important because this is the layer of cells that will form the 3 primary germ layers, the ectoderm, mesoderm, and endoderm. And all tissues in your body are derived from these 3 primary germ layers. So now let's move on to the embryonic stage. Now remember, this begins around day 16, and what marks the beginning of the embryonic stage is the completion of a process called gastrulation. Gastrulation is the process that forms the 3 germ layers in the embryo from the epiblast. The 3 germ layers that form in the embryo are called the ectoderm, mesoderm, and endoderm. Now, this is important because each germ layer becomes a different type of tissue in the human body, so in some ways their fate is being decided at this point. And how exactly do these germ layers form? Well, it's through a process called invagination. So remember, we have this flattened disc of cells with the epiblast and the hypoblast on the bottom here. You have invagination of the epiblast cells, so this groove forms, and some of the epiblast cells move into the groove. And during the first invagination, the epiblast cells actually replace the hypoblast, and those cells become the endoderm. And then there's a second invagination of epiblast cells, so they move in. And those cells form a middle layer called the mesoderm. So I always remember M for middle. Endo means inside. So we have the endoderm, the mesoderm, and the epiblast cells that remain behind become what we call the ectoderm, or the outer layer of cells. Each germ layer is destined to become a certain type of tissue, and you need to know what tissue types develop from each germ layer. And we could actually list a whole bunch more, but the only ones I want you to know are what is shown in this diagram here. So remember, the endoderm layer, this is the innermost layer, so it's going to become a lot of the internal organs, like the digestive system and accessory digestive organs like the liver and pancreas. And it also will form the lungs as well. The mesoderm layer, this is the middle layer, it becomes the major structures of the circulatory system like the blood vessels and heart. It becomes part of the kidneys, as well as the structures that make up the skeletal system like the bones and ligaments, and also the structures that make up the muscular system such as the smooth muscles and skeletal muscles. The ectoderm, this is the outermost layer. So I always remember that it's going to form anything on the outside of your body as well as things that help to detect your outside world. So these would include things like your hair, nails, skin, and also your nervous system, because your nervous system helps you detect the world around you. Around week 4 of embryonic development, the embryo is no longer a flattened disc, and it actually starts to fold in on itself. And this embryonic folding is extremely important because it starts to create a cephalic end, or a head end, and a caudal end, or a rear end. So here we can see the flattened disc. This is a longitudinal view. So this is the embryo here, and you can see it's been sliced longitudinally. And here you can see folding on this end and this end, so the two ends are curving in on each other here. And as it folds more and more, this end is becoming the head, and this end's becoming the tail. Here you can see the heart starting to develop. And also surrounding the embryo, you see a bunch of fluid-filled cavities forming. On this end, we have what's called the amniotic cavity. Here, we can see that it's folding around this cavity here called the yolk sac. And here we have a little pocket coming off of the yolk sac called the allantois. And we'll talk about the function of these structures in a little bit. So after the germ layers have formed and the embryo has started to fold in on itself, then we have a process called organogenesis, which is when the rudimentary organs are formed within the embryo. Now remember, these organs are still not functional. They still have to go through a lot more development and maturation before the embryo could survive outside of the mother's body. But here are some images just to kind of show you what's happening here. This is week 3 of development. We can see the formation of the neural plate and neural groove, which are all going to become parts of the nervous system. This is week 4, and at week 4 you can see the eye, the lens of the eye starting to form here. Here are some pharyngeal arches developing, which are characteristic of chordates. These will eventually disappear. Here we have the heart forming, an arm bud, and a little leg bud forming, and there's still a little tail there. This is at the end of week 7. Here you can see the eyes, the ears forming, the digital arrays, and the hand, and you can see the legs are starting to form a little bit more. This bulge right here is actually the liver forming in the inside. By week 8, the embryo starts to look like a human from the outside. And so here you can see an embryo at 8 weeks of development. Here's the umbilical cord. Here's the amniotic sac. Here's the yolk sac connected. You can see a little string connecting it here to the umbilical cord. Although this little embryo looks very human-like, it's only 3 centimeters long, so it's the size of this tiny little cashew here. That would be the size of an 8-week embryo, 3 centimeters. Now let's look at the accessory organs that develop alongside the embryo. These include extraembryonic membranes, including the amnion, which you can see here, outlined in blue, the yolk sac, shown in yellow, allantois, shown in green, and the chorion, shown in pink here. We're going to talk about the function of these extraembryonic membranes in the next slide. Another accessory organ that develops alongside the embryo is the placenta, which is shown down here. Here you can see the maternal part of the placenta. What's shown in pink is the fetal part of the placenta, and it's connected to the embryo through the umbilical cord. So let's look at the functions of the extraembryonic structures, and you want to know the general functions of each one of these structures. So let's start with the amnion. The amnion is a membrane that's filled with amniotic fluid, and this is shown in blue in this diagram again, the amniotic fluid. The function of the amnion and the amniotic fluid is to surround and protect the developing fetus. At the beginning of development, the amniotic fluid in here is actually formed from filtration of the maternal blood. However, eventually it starts to be produced by actually the fetus urinating and generating more of this amniotic fluid. Another role of the amniotic fluid is that later on in development, the fetus will actually start to inhale and swallow the amniotic fluid, and that actually helps to stimulate their lung development. Whenever a mother's water breaks, it's actually the amniotic sac here that's breaking and tearing open, causing fluid to come out. The next structure that we're going to look at is the yolk sac, shown in yellow here. It contributes to the formation of the digestive tract and also has 2 other major functions. It is the source of the first blood cells, and it's also the source of the primordial germ cells. And these are the cells which are going to become sperm and egg. So they actually form within the yolk sac before they make their way into the gonads. Next, we have the allantois. This is an out pocket of the yolk sac, shown in green here. It forms the foundation of the umbilical cord along with the yolk sac, as you can see in this image. It will become part of the urinary bladder. And usually whenever the fetus is born, the allantois, most of it has disappeared at that point. However, there are cases where part of it is still attached to the small intestine, and this is called Meckel's diverticulum. And so here you can see a remnant of the allantois attached to the small intestine. This can cause a lot of digestive problems because food and stuff can get stuck in here, and that can cause bacterial infection. This can also cause hemorrhaging or bleeding, where you'll see a lot of blood in the stool as well. So this has to be corrected if this does occur in an individual. Now, the last extraembryonic structure that you should know is known as the chorion. And if you remember, this is derived from the trophoblast of the blastocyst. This is the outermost membrane. It's shown in pink here. And it surrounds all the other extraembryonic structures. And it becomes a fetal part of the placenta. And you can see that as these little pink branches coming off here where the placenta would be. So exchange of nutrients and waste occurs between the maternal portion of the placenta and the fetal portion of the placenta. So now let's briefly talk about what characterizes the fetal period of development. Now remember, this begins at week 9 to birth. One of the characteristics of this stage is maturation of the tissues and organs to a point where the infant could actually survive outside of the mother's womb. Now remember, the earliest that an infant has a chance to survive outside the mother's body is 22 weeks after fertilization, or in other words, 24 weeks after her last period. So at this point, if an infant is born, it's considered premature and it requires a high amount of intensive care. Also associated with the fetal period is rapid growth of the fetus. Most of the weight gain that occurs during pregnancy is during the last 2 months of pregnancy, because that is when the fetus is growing at the fastest rate. Here you can see a fetus at week 12. On the right, you can see a fetus at week 20. So this fetus still would not be viable outside of the mother's body. Here is the placenta, the umbilical cord. Here's the amniotic sac, and this is actually the uterus out here. Here is showing how the fetus develops over the last few weeks of development. And you can see how they keep gaining more and more weight. The head becomes more proportional to the body as development occurs. So now let's look at what's happening inside the mother whenever the fetus has reached full term, so at 38 weeks after fertilization. So here you can see there's just not much room left for that fetus to grow anymore. The uterus has expanded as much as it can. Here you can actually see the ovaries dangling off the sides of the uterus from these ligaments. You can see that the small intestines are completely squished, and also the bladder down here is being completely squished as well. If we look here, we can see what the internal anatomy of the mother would look like before she got pregnant. The intestines would be taking up this whole space here. Here we have the bladder. Here's the uterus right here. Here's the stomach. However, after the baby is full term, here you can see the baby right here with the placenta attached to the uterus. And you can see all the intestines now are getting squished. And look at the bladder here. So this explains why mothers usually have to go to the bathroom pretty frequently, because the bladder can't really expand to hold a whole lot of urine. And also, it's harder for the mother to eat large meals because the stomach is getting squished as well. Pregnancy is also associated sometimes with constipation because it's really hard for food to move through the intestines because of how squished they are. Next, we're going to talk about prenatal nutrition. So during gestation, the conceptus is nourished 3 different ways. First, the conceptus is fed with uterine milk. Uterine milk is produced through uterine secretions that feed the conceptus until it actually implants. So this is from fertilization until about day 5 or 6. After the conceptus starts to implant, it's fed through something called trophoblastic nutrition. So remember, when we look at the blastocyst, this outer layer of cells is called the trophoblast. And during implantation and until the placenta develops fully, the conceptus is initially fed through these trophoblast cells. And these trophoblast cells will actually absorb nutrients from specialized cells in the endometrium as it implants into the endometrium. And trophoblastic nutrition will continue from week 1, whenever the blastocyst starts to implant, until about week 12, when the placenta fully takes over. The placenta begins feeding the conceptus around week 4. However, it's being supplemented by trophoblastic nutrition. The placenta doesn't completely take over nourishing the fetus until about week 12, whenever it has fully formed. This diagram here is showing the types of prenatal nutrition that are occurring the weeks after implantation. And you can see during the first couple of weeks after implantation, trophoblastic nutrition is the primary means by which the conceptus is getting nutrition. So remember, those trophoblast cells are absorbing nutrients directly from the endometrium. The placenta starts providing some nutrition early on after implantation, but doesn't completely take over until about week 12 after implantation, where then the conceptus is pretty much getting all of its nutrition from the placenta itself. Now let's talk about the placenta in more detail. The placenta is a disc-shaped organ that adheres to the uterine wall on one side, and it adheres to the fetus on the other side via the umbilical cord. The placenta is formed from both maternal and fetal tissue, and I'll talk a little bit more about how this is structured in the next couple slides. Let's list some of the functions of the placenta. First, the placenta provides the fetus with oxygen and nutrients. Second, it removes metabolic waste and carbon dioxide. It also produces estrogen and progesterone, which is important in maintaining and building up the uterine lining throughout pregnancy. The placenta also secretes other hormones that are important in pregnancy, such as relaxin, which relaxes the ligaments in the pelvis and also softens and dilates the cervix prior to labor. It secretes the hormone HCG, which stands for human chorionic gonadotropin, which is released early on in pregnancy, and this actually keeps the corpus luteum around within the ovaries. The corpus luteum produces progesterone and estrogen until the placenta can take over around week 12 of pregnancy. The placenta also produces HCS, which stands for human chorionic somatomamotropin, and this hormone is really important in modifying the metabolism of the mother to provide more nutrients for the developing fetus. And finally, one of the major functions of the placenta is to transfer maternal antibodies to the fetus, and this provides passive immunity to the fetus during the early stages of development and even after the baby is born. So again, here this illustrates the importance of the HCG hormone. So the HCG hormone is actually produced by the fetal part of the placenta early on in the weeks of pregnancy. And HCG functions in keeping the corpus luteum around because this is the primary producer of progesterone and estrogen in the early stages of pregnancy. However, around week 12, the placenta takes over the production of progesterone and estrogen, so HCG levels drop. The corpus luteum degenerates, and the placenta now just takes over and the production of this estrogen and progesterone. Again, HCG is the hormone that is detected by pregnancy tests because of its large peak in the amount of it in the body early on in pregnancy. Let's look a little bit at the anatomy of the placenta. So the placenta is formed from fetal and maternal tissue. If we look at the maternal tissue that makes up the placenta, it's the functional layer of the endometrium. So here you can see the functional layer of the endometrium, so the inner lining of the uterus, and you can see that it forms these spaces that are pools of blood that contain maternal blood here. The fetal part of the placenta is formed by the chorion membrane, which again is formed from the trophoblast of the blastocyst. And so here's the chorion here, and you can see that it creates these branches going into, that kind of branch into like roots into the functional layer of the endometrium. And these branches are called the chorion villi, and each one contains blood vessels. And so what can happen then is nutrients can leave the mother's blood and enter the blood of the fetus. And waste and stuff can exit the blood of the fetus and enter the blood of the mother. An important thing to note here is that fetal blood and maternal blood do not mix at all, but they do come in very close contact. So there is a very small barrier between the fetal blood and the maternal blood, and this allows the fusion of nutrients and waste to go back and forth, but prevents the blood from actually mixing. How does the nutrients get to the fetus, and how does the waste leave? Well, that's all through the umbilical cord. So the umbilical cord actually consists of one umbilical vein that feeds the embryo with nutrients and oxygen, and you can see that here. And it's shown in red because it's oxygenated. And you can remember that the umbilical vein is supplying the fetus with nutrients if you remember that veins always carry blood towards the heart. So they're carrying blood towards the heart of the fetus. Whereas the umbilical arteries are actually paired, and they are carrying waste away from the fetus. And if you remember, arteries always carry blood away from the heart. So I always remember it as carrying blood away from the fetus. And so it's carrying the deoxygenated carbon dioxide rich blood out here, and as well as waste out here, so that the mother can take up that waste and stuff and then remove it from her own body. Here's a close-up of the placenta. So we can actually see the umbilical cord here. Here is the placenta over here. If we zoom in, we can see the umbilical vein shown in red and the umbilical artery shown in blue. These branch into much smaller vessels in the chorionic villi, these little branches. And here is the maternal blood supply surrounding the chorionic villi, and exchange of nutrients and waste are happening between the small barrier here. The placenta is actually expelled after birth, and this is what we call the afterbirth. So this is what it would look like after it's expelled from the mother. And you can actually see this is the fetal side of the placenta, so it's really smooth. So the fetus was actually living inside this part of the placenta, and the amniotic sac was wrapped around it. And here's the umbilical cord right here. This is the side of the placenta that was actually attached to the uterus. So it was actually adhered to the uterus, where the blood supply was very, very rich. And so it actually separates from the uterus right after birth. The final thing that I'm going to talk about in this lecture video are the 3 stages of labor. And what I want you to know here are just what are the characteristics of each stage of labor. So stage 1 of labor is characterized by thinning and opening of the cervix. Thinning of the cervix, the technical term for that is effacement. And as the cervix starts to open, we say it's dilating. Now this coincides with contractions that are occurring in the uterus. And these contractions will actually start off kind of far apart from each other, and then they'll become more and more frequent to a point where each contraction is happening about 4 minutes apart. And the duration of each contraction will get longer to where it's happening about 1 minute apart. And whenever it's about 4 minutes apart, each contraction, and the duration is about 1 minute, and it lasts for about an hour, that's usually when they say you need to try to get to the hospital then. So initially the cervix is not effaced or dilated. However, these contractions help to soften or thin out the cervix, and then also help it to become dilated. As you can see here, the cervix is thinner, and now it's starting to dilate. A full dilation of the cervix is 10 centimeters, and this is how wide it has to be to allow the baby's head to get through. And this is the top of a cashew container. This is about 10 centimeters across, so you can see how much the cervix must dilate in order to deliver the baby. After the cervix is fully dilated, then we enter stage 2 of labor, wherever the mother can start pushing and when a birth actually occurs. And this can take anywhere from like 20 minutes to over 3 hours for pushing out the baby. And so this begins with presentation of the head. And usually the face is pointing down. So this is the mother's stomach here, and the baby's face is pointing down. There is something called back labor, where the baby's actually facing up. And this actually can be really, really painful for the mother, because it's harder to get the baby out when it's facing up that way. Then the baby rotates, and then it comes out the whole way, and you can see the umbilical cord is still attached to the placenta, which is still adhered to the uterus at this point. One thing that they do immediately after birth now is something called skin-to-skin contact, where they take the baby and they put the baby right onto the skin of the mother. And the reason that they do this is because studies have shown that the baby can adjust easier to life outside the womb whenever they have this skin-to-skin contact. So it helps them regulate their breathing, it helps them regulate their body temperature quicker, and it also helps them regulate their blood glucose levels easier. In addition to that, usually the baby will actually start to breastfeed within an hour after being delivered, once they've had this skin-to-skin contact. After the birth of the baby is stage 3 of labor, which is delivery of the placenta, or the afterbirth. This usually takes about 5 to 20 minutes after the baby is born, and only lasts a few minutes, where the placenta detaches from the uterus and is expelled through the vagina. And If you're curious of what the placenta actually looks like after it's expelled, this is a video that actually shows what the placenta looks like. And it's kind of interesting because they actually show the texture of the umbilical cord, which is a very unique structure. So if you're not squeamish and you're willing to watch something like this, I'll put a link to this video of the afterbirth below. All right, so this is the end of part 1 of the human development and heredity chapter. Video 2 will look at heredity and genetics. We are making our first trip to triage. I don't think this is going to be our last trip. I'm hoping they send us home because I'm not having any contractions yet. But Eddie's driving to the hospital. Hello. We're still doing good. It's 9:05. I'm still feeling some contractions. They're getting a little stronger. And my Yumi's moving around like crazy.
She moves away from the monitor, so there's these like blank spots because she moves away from it. And you hear the contractions in the spaces between them. It's almost 5:30, and we're in the process of going into the labor and delivery.
We got moved to labor and delivery around like 6 a.m. or so. Right?
Yeah, it was, let's see, the moving process was about like 5:30.
Maybe 5:30.
Yeah, it was like 5:30.
Because I was getting really strong contractions. And
yeah, she was a champ.
It was like, I think I was 6 when we left there.
Yeah.
And then by the time we got here, I was like 8 something
centimeters.
But, yeah, I got the epidural because I've never felt pain that intense.